Chapter 17, Section 2, Oxidation of Fatty Acids, Part A. This is a long section of the chapter, so it's split into 2 videos, 2 lectures, okay? So don't freak out. So, stage 1 of beta oxidation. Gotta add Acyl Coa. Okay, makes sense. Acyl Coa is going to be added to the carboxyl end. Okay, so this carboxyl end, and then we number from here 1, 2, 3, 4, 5, 6, 7, 8. C1, C2, C3, C4. And then we're going to cut between C2, C3. Cut. New acetyl CoA is going to be added. C1 now becomes C1, C2. Cut. Add another CoA. C1, C2. Cut. This is what beta oxidation is. By cutting every 2 carbons and having it already attached to an acetyl group, I'm sorry, not to an acetyl group, to a CoA group, it forms acetyl CoA. Acetyl CoA that can be fed directly into the citric acid cycle. So our 16 carbon length fatty acid is going to yield 8 acetyl CoAs, which are going to go through the cycles. It's going to result in 16 carbon dioxides and 64 excited electrons in the form of NADH or FADH2. The NADH, FADH2s are going to go to the electron transport chain of the respiratory cycle of oxidative phosphorylation. Energy is going to be bled off, final electron except for oxygen, giving you water, and then a buttload of ATPs. So how does this work? Well, 4 basic steps. First step, add a CoA. Yeah, I know that sounds like, ooh, but yeah, first we got to get to a CoA. Acyl Coa Dehydrogenase. So once you have the CoA, well, we got to do some work. Alpha, beta. We're going to cut here. Boom. They call this the trans delta 2 enol CoA. So we're taking this carbonyl, and we're going to take this CH2, and we're going to put a double bond here between these 2 carbons. To do so, we need to remove 2 hydrogens. Hey, guess what? Those hydrogens are going to have some energy in those electrons. So we're going to take FAD, and we're going to reduce to FADH2. And those 2 electrons are going to go to the respiratory chain. So now we have this double bond oxygen. Carbonyl, carbon, carbon double bond. We're going to see that Acyl Coa Dehydrogenase, there's quite a few isoenzymes for this. Remember, isoenzymes are enzymes that do roughly the same thing, but their substrates are different. VLCAD, MCAD, SCAD. These are in the mitochondrial matrix, and that's associated with the inner membrane of the mitochondria. A lot of that depends on what the fatty acid chain length is, how many carbons are there. The electron transfer flavoprotein, ETF. ETF is what's going to accept the excited electrons from FADH2. You see this as the ETF ubiquinone oxidoreductase. This is part of that electron transport chain associated with what we call respiratory, the respiratory chain, the oxidative phosphorylation step that's at the mitochondrial cristae. It's going to take these excited electrons, pass them on to the cytochromes and things like that. So you can start bleeding off energy. We see this whole process, the Acyl Coa Dehydrogenase, is very much like that succinate dehydrogenase that we talked about in the citric acid cycle. Remember the one that had the E1, the E2, the E3, that when you looked at the overall structure, the cluster of those complexes, it looked like the flowering plant thing. Yes, we see roughly the same thing here. You're going to have enzymes bound to an inner membrane, double bond introduced into carboxylic acid between the alpha and beta carbons. Going to strip off some hydrogens. They're going to go into the respiratory chain later. That enol CoA hydrolate, enol CoA trans delta 2 enol CoA is now going to have to be modified back. So you're going to use the enol CoA hydratase that's going to add water. So this carbon 3 is now going to get a hydroxyl. Carbon 2 is now going to get a hydrogen back. This is L-beta hydroxy acyl CoA. Then you're going to strip off some hydrogens, hence a dehydrogenase, NAD to NADH, and you're going to be stripping off the hydrogens off of carbon 3, forming that carbonyl. Why do you want to have a carbonyl there? Well, because remember you got this carbonyl here at carbon 1. At some point in time, you're going to want to do a cut right there. What was carbon 1 here? Carbon 3 here? Well, this is now going to be carbon 1, and everything we just went over is going to repeat all over again. These electrons are going to be entering into the respiratory chain, the electron transport chain, via the NADH dehydrogenase, complex 1. It's going to take those and it's going to start passing them along and bleeding off the energy. Accepts electrons from the NADH formed in the beta hydroxyacyl CoA dehydrogenase reaction. So basically what this is saying is the NADs here, the NADHs, have their own target within the cytochrome complex. This is new to me because I was always taught that NADs is NADs is NADs, and once they become NADH, there's little to no difference between NADHs made in the cytoplasm or to the mitochondria from the citric acid cycle or here with beta oxidation. We'll see. Acyl CoA acetyltransferase, also known as thiolase. Oh, now we're getting somewhere. So we have the CoA. Here's our sulfur carbon bond. Carbonyl, CH2, carbonyl, CH2. All right. Acyl CoA acetyltransferase, acetyl group transferase. So you're cutting. You're doing the cut right here. That's what this bad boy's doing. It's cutting and releasing. Here is acetyl CoA. And here, for all intents and purposes, is literally the same thing we just started with these 4 steps with. It's just now, instead of C16, we're now at C14. We have what is referred to as TFP, Tri-Functional Protein. Tri-Functional Protein is a multi-enzyme complex. Multi-enzyme complex that's at the inner mitochondrial membrane. Catalyzes steps 2 through 4 that we just talked about of the beta oxidation pathway. But this is for the large or long chain fatty acids. This TFP protein is associated there because, hey, look at this. We've seen this before. Substrate channeling. Substrate channeling is when you have a protein or a coenzyme A or a coenzyme, sorry, not coenzyme A, a coenzyme that's going to grab something and it's going to hold it and it's going to move it from one enzyme to the next, to the next, to the next. It is literally acting as a delivery service. So that way, this enzyme substrate, which is going to be the next enzyme's, this enzyme's product, which is the next enzyme substrate, we don't have to wait for it to slowly diffuse from this enzyme to that one. It's going to be directly delivered where it needs to be. This is what they call a heterooctamer. Hetero, different proteins. Octamer means it's 8. 4 alphas, 4 beta subunits. The alpha subunits contain, what is it, enol CoA hydratase. The betas, hydroxyacyl Coa Dehydrogenase. These are the 2 enzymes that we've seen within steps 2 through 4. What's the logic behind this? Well, first, it's stable to go through, do everything this way. Okay? If you have single bonds, okay, so there's no double bonds, so this is all saturated fatty acids. We can go through and things can be cut. They talk about in the textbook the ketone function of the beta carbon, the C3. Okay? How it makes a good target for nucleophilic attack. So, carbonyl, CH2, carbonyl, which is the beta carbon. It allows for the nucleophilic attack. It allows for the incoming CoA to have something to bind to. So when you have the terminal coenzyme A, sulfur, this is that carbonyl, here's a CH2, and this is where the cut is going to be, so this is going to become CH3. This allows for a good nucleophilic attack that's going to be the new coenzyme A coming into this open bond for the remaining fatty acid chain. When you look at it, this is a pattern we've seen before. This is a conserved sequence. This chain of events. Here's the fatty acid with the coenzyme A. Here's the carbonyl, CH2, CH2. Come in, remove some hydrogens, double bond, add water back. Now you have a hydroxyl and you've replaced the hydrogen. Strip off the hydrogen from the hydroxyl, NADH. You now have a carbonyl. We see this with beta oxidation. We saw this at the citric acid cycle as you went through and succinate to oxaloacetate. We see this in amino acid synthesis. In getting the amino acid, catabolism, getting it ready to enter into the citric acid cycle and whatnot. So this is something we've seen before. The enzymes here, the process, the overall pattern is nothing new. What's new is the names of the enzymes and the individual intermediates. But the overall pattern is stuff that has been talked about and lectured on in this course already. Because of the way this was working, coenzyme A attaches to the carboxy terminal end. Go through those 4 steps that we just talked about. Cut, add a coenzyme A to the new carboxyl terminal end. Go through those 4 steps. Cut, add the CoA. So you go from C4, C12, C10, C8, C6, C4, and once you cut the C4 in half, you end up with 2 acetyl CoAs. All of these acetyl CoAs are readily available to enter into the citric acid cycle. So every pass, every time it goes through that beta oxidation, you're removing 1 acetyl group. 2 pairs of electrons, 4 hydrogens, 4 protons. I just broke my thing. There it is. So if you're looking at palmityl CoA plus CoA plus FAD, NAD, and water, you end with myricetyl CoA, acetyl CoA, FADH2, NADH2, NADH, and hydrogen. That's 1 round. Then you go and you cut the myricetyl and do this all over again. Next round, do it again. Next round. So if you start with palmityl CoA, you will end with 8 acetyl CoAs, 7 FADH2s, 7 NADHs, and 7 free hydrogens. Okay, the electron transport chain. That's where the FADH2s and NADHs are going. Each FADH2 donates a pair of electrons, 2, to the electron transport. Okay, remember from previous chapter, since the FADH2 is donating its electrons, not at the beginning, but at near the midpoint of the electron transport chain, it's going to generate fewer molecules of ATP than the NADH that starts at the first step of the electron transport chain. Less energy gets bled off, less hydrogens get pumped across. More energy gets bled off, more hydrogens pumped, which means there's more chemiosmosis for the turning of ATP synthase, generating more ATP. So when it comes down to it, each time a fatty acid goes through beta oxidation, 4 ATP. So going from 14 to 12, there's 4 ATP that can be made. Going from 12 to 10, 4 more. 10 to 8, 4 more. One of the side notes of the textbook talks about this. That because of those electrons that are being transported through the electron transport chain, and that final electron acceptor being oxygen, well, the oxygen just doesn't take the electrons. It takes the electrons, some of the free hydrogens, and the electrons, and yields water. They call that metabolic water. This is water different than the water you ingest. This is water that's a byproduct of an enzymatic reaction. So you end up with a product of water. This is how certain animals can hibernate. They store up lots of fat. You think, they're showing bears here. Think about it, bears. Late summer going into fall are going to consume anything and everything they can. Build up those fat stores so that when they go into hibernation, those long sleep pattern cycles, they don't need water. They're not going to dehydrate, and die because, well, here's the water they're going to be making as they're breaking down those fat stores. This is also how camels. Okay, I hate to tell you, camel humps not for storing water. The camel humps for storing fat. Fat triglycerides that are going to be broken down, beta oxidation, get those excited electrons, make some metabolic water. So we see this here too. Palmitoyl-CoA, bunch of CoAs, oxygens, inorganic phosphates, ADP. You get 8 acetyl-CoAs, 28 ATP. This is that substrate level phosphorylation in the citric acid cycle. And a bunch of water.